My name is Lucas Sasaya, and I'm a bioinformatics summer trainee in the Myon lab. And I'll be presenting on the CFDE Chatbot Assistant. First, to put things into context, I would like to introduce the Common Fund Data Ecosystem, or the CFDE. The Common Fund Data Ecosystem is a relatively new NIH Common Fund program that was initiated to create an ecosystem to enhance data sharing, interoperability, and accessibility to Common Fund datasets hosted by Common Fund's programs, Data Coordinating Centers, or DCCs. Some of these are listed here. The CFD Workbench, currently being developed by the CFDE Data Resource Center, headed by the Maya Lab at the Icahn School of Medicine and the Subramaniam Lab at UCSD, is tasked with creating two portals, an information portal and a data portal for the CFDE. This is the landing page for the information portal and the various sections it covers. The data portal has a search engine, a data matrix, listing of use cases, tools and workflows, data and metadata submission system, and a chatbot, which will be the topic of this presentation. Before describing the chatbot, I would like to acknowledge Giacomo Marino, a bioinformatics software engineer at the Maya Lab, who develops the chatbot. My role in the project was to add additional functionality to it. The CFD chatbot can do the following. It can answer queries related to DCC datasets and general questions related to the CFDE and the DCCs. The interface has text chips that are example queries to guide users about the information they can obtain from the chatbot. The interface also has icons of each Common Fund program, and once these icons are clicked, the user receives textual information about the program. The chatbot interface also has support for text-to-speech and speech-to-text. One of the main purposes and features of the chatbot is to enable users to ask questions naturally and flexibly while invoking accurate tools and responses. And the chatbot is currently available at this URL. Now let's look under the hood to learn how the chatbot operates. We are using OpenAI assistants to perform several tasks, including function calls and information retrieval. Importantly, the chatbot only responds to specific CFDE-related queries. The workflow starts with a query that is sent to the OpenAI assistant. It is then determined if we need to perform information retrieval or invoke a function call. For function calls, the assistant uses the Playbook Workflow Builder API or accesses the CFDE DCC API directly. The response is then processed and displayed on the chatbot frontend and returned to the user. Here's an example of a previously implemented workflow for the query, which L1000 drugs most significantly up or downregulate the expression of the gene STAT3. The query is sent to the OpenAI assistant, which calls the Playbook Workflow Builder API with parameters for a link's L1000 reverse search. The results are returned, visualizing the chatbot frontend, and include a button to open the results on the Playbook Workflow Builder site. For the updated version of the chatbot, I added 4 new input types, 4 new processes from 7 new processes from 6 Common Fund DCCs. Ultimately, these new processes were added to direct users to existing Common Fund programs' data portals. One of the processes that I added is for GlyGen. The GlyGen data portal contains over 53,000 glycans and 138,000 proteins from 12 different organisms. The goal of this Common Fund program is to improve our understanding of the structural and functional roles of glycans in health and disease. Using the chatbot, you can now perform a biomarker search similar to the way such search can be performed directly from the GlyGen portal. The advantage of using the chatbot is that you can interface with it directly by complete questions about biomarkers. You can start by using the example chip or type in a related question that matches your interests. Here we see the response. It includes the biomarker, assessed biomarker entity, and the condition. Here too, you can open the hyperlinked IDs and view the results on the GlyGen portal. The results are also downloadable as JSON or CSV files. The next process I added is from the Human Biomolecular Atlas program, or HuBMAP. The goal of the HuBMAP program is to develop an open and global platform to map healthy cells in the human body at the single-cell level. This includes developing tools to integrate and map diverse single-cell data for better understanding the organization of cells in human tissues. The process I added to the CFDE chatbot is similar to the HuBMAP dataset search feature on the HuBMAP portal. After using a query similar to the following text chip, the chatbot will parse the necessary information related to the tissue, assay, and group name for the dataset search. Following this, it will return a table of relevant datasets, which include the dataset IDs, titles, and group names for each entry. While viewing the data, you can open any of the hyperlinked HuBMAP IDs in the HuBMAP portal. The next process I added is from the 4D Nucleome data portal. The 4DN data portal has numerous datasets, software packages, and descriptions of protocols and reagents related to research on chromatin or structure and organization. The goal of the 4D Nucleome Common Fund program is to understand how changes in nuclear organization over space and time impact gene expression and cellular function, and the effects of such changes on normal physiology and in disease. The 4DN data portal has a well-designed data browser with multiple filters that facilitate identifying specific datasets generated by the consortium. The CFDE chatbot now answers queries similar to those available from the 4DN dataset browser. After using a query similar to the following text chip, the chatbot responds with filters for organism and experiment types. The input field can be used to search for specific values, and the user can select multiple options provided for each filter. The CFDE chatbot then displays an output that includes the hyperlinked title of the dataset, description, and a button to open the complete results in the 4DN data portal. The final process that I added is from the Molecular Transducers of Physical Activity Consortium, or MoTrPAC. The MoTrPAC consortium aims to study the molecular changes that occur in different tissues due to exercise. So far, the consortium published a large animal study where multiomics data was collected from 18 different tissues before and after endurance exercise training. The MoTrPAC Data Hub contains a differential abundance search engine for genes, proteins, or metabolites, and a way to view these changes at each time point. Users can also perform the search with the chatbot by using a query similar to this text chip. The chatbot first detects the key term the user provides. Then it opens with a dialog box with a few input options to verify that the query was understood correctly. The second image shows the gene input field from the previous slide that includes tooltips and filters for tissue, assay, and p-value. After submitting the query with the selected options, this table provides a summary view of the results. Some future direction for this project include implementing the processes that I described as playbook metanodes, adding folders for chips based on input and output type, enabling users to ask questions related to multiple DCCs and workflows, creating an ecosystem of chatbots that interact with each other, and adding more detailed documentation about Common Fund programs, CFD partnerships, and CFD training and outreach activities such as this summer program. I'd like to thank all the members in the Mayan Lab for their guidance and support. I'd like to especially acknowledge Giacomo Marino and Dr. Mayan for their direct mentorship throughout this program. Finally, I want to acknowledge Ms. Jenkins for making this program possible. Thank you.